Hi, my name is Serhat Gumurtchi from Seraph Research Institute, and I'm excited to present this work on behalf of my team and my co-authors. A novel COVID-19 cure strategy, hijacking SARS-CoV-2 RNA-dependent RNA polymerase. As you know, SARS-CoV-2 is a single-stranded positive RNA virus that utilizes negative-sense subgenomic RNA intermediates for its replication and viral protein synthesis. The image on top represents how nuclear protein is produced by our DRP using a negative strand subgenomic RNA as a template to make mRNAs. Through this mechanism of action, we designed an oligonucleotide called Hijack RNA that looks like a subgenomic negative strand RNA to the viral polymerase, and it hijacks the polymerase to produce a suicide molecule, diphtheria toxin fragment A. DTA kills cells through inhibiting protein synthesis, and because it has no membrane penetration abilities, it cannot enter other cells, and it is non-toxic when it's released from a dead cell. To run our in vitro studies, we have packaged our novel vector in AAV particles and ran killing assays on infected or uninfected Cali3, Vero, and HipG2 cell lines, as well as uninfected Jurkat, HGK, and BHK21 cell lines. In our in vivo studies, we have developed infected cell line xenotransplant models on SCID mice or actual infection models with human ACE2 transgenic mice using luciferase expressing recombinant SARS-CoV-2 particles. In our in vitro studies, we have demonstrated that hijack RNA expression has no effect on uninfected cells, and it kills all the infected cells in vitro within 48 hours. If you look at the graph on the left over here, you can see the growth curve of untreated and test AAV treated uninfected cells, and they're essentially the same. So we have no effect on uninfected cells with our vector. And graph on the right shows infected cell virocultures where test AAV is administered on day 2, 48 hours after cell culture started. And within 48 hours AAV administration, all the infected cells were essentially dead in the cell culture. If you look at the right side of the slide, we see representative pictures from our in vivo studies where the test AAV eradicated all the infected cells within 6 days after test AAV administration in the infected mouse model. And we have seen no treatment-related toxicity, either acute or chronic, within 2 months after AAV administration or 2 months after virus clearance. And we have seen no relapse of infection within this period. The mice are still being kept alive, so we will have month 3 sacrifices to look at long-term toxicities in other organs. We have thus concluded, after our studies, our novel hijack RNA expression eradicated SARS-CoV-2 infection in vitro within 48 hours and in vivo within 6 days. Novel hijack RNA had no effect on uninfected cells, and the treatment had no acute toxicity in mice. No emergence of virus or treatment-related toxicity were observed as of 5 weeks after virus clearance. Because AAV persists in non-dividing cells, an aerosolized treatment targeting respiratory epithelial cells could actually provide a long-term, durable, and easy-to-administer pre- or post-exposure prophylaxis simply by waiting in healthy cells and effectively ambushing the virus immediately upon transmission. An effective protection could last up to 1 year, which is the approximate turnover of lung epithelial cells. And given the conserved nature of UTI regions of most human coronaviruses, this approach would work against SARS-CoV-1 as well as some of the other common cold coronaviruses. A hijack RNA approach also could be useful for other infections like hepatitis B, HIV, and influenza. In my lab, we have done several studies targeting hepatitis B and influenza, and we presented these in the past 2 years. So if you're interested in looking at those, the links to those presentations are in the PDF handouts. I would like to thank you all for listening to my presentation, and I would like to thank my lab members as well as my friends and collaborators in other institutions for making this project possible. Thank you.